The Cell Membrane by Elizabeth, Eric, and Kara. Phospholipids consist of a phosphate group and two fatty acids attached to a glycerol molecule by a dehydration reaction. The head, which contains the phosphate group, is hydrophilic, whereas the tails, which consist of fatty acids, are hydrophobic. In an aqueous external environment, phospholipids arrange themselves so that the hydrophobic portions avoid water while the hydrophilic portions face water. As a result, micelles can form. Additionally, in the case of cell membranes, which are in contact with an aqueous cytoplasm, these molecules form a phospholipid bilayer, with the heads facing out and the tails facing in. In a plasma membrane, there are integral proteins, such as carrier proteins and channel proteins, that extend through both layers of the membrane. These have portions with hydrophilic R groups near the ends of the protein, whereas the inner portions near the tails of the phospholipids contain hydrophobic R groups. Membranes also have surface proteins that probably have hydrophilic R groups, which attach to the membrane using hydrogen bonds, dipole interactions, or ionic bonds. The model used for the structure of a membrane is the fluid mosaic model. This describes the proteins as a mosaic and the phospholipid bilayer as the fluid they are bobbing in. The membrane can be stabilized with cholesterol, a steroid that lowers fluidity at high temperatures and increases fluidity at low temperatures. Embedded proteins and surface proteins serve many purposes. They can help the cell exchange nutrients with its environment, as in channel proteins, catalyze reactions, or help the cell move. Some have polysaccharide chains attached to them. These are called glycoproteins, and they are like identification tags for cells. Because the majority of the plasma membrane is constructed of nonpolar hydrocarbon chains, selective permeability is mostly determined by the size and polarity of the molecule. Nonpolar molecules typically pass through the semi-permeable membrane better than polar molecules and ions, and small molecules typically pass through the membrane better than large molecules. A small, nonpolar molecule like CO2 and O2 can pass freely across the membrane. Glucose, a large and polar molecule, cannot pass through the membrane and requires proteins to facilitate diffusion. The proteins either provide a channel to allow diffusion or change shape to change their affinity for certain molecules. Diffusion of water is called osmosis. While water can cross the cell membrane, cells often have a channel protein called an aquaporin that allows 3 billion water molecules to diffuse per second. The cell wall is the protective semi-permeable outer layer of a plant cell. A major function of the cell wall is to give the cell strength and structure and to filter molecules that pass in and out of the cell. It prevents the cell from bursting if too much water tries to enter the cell. Molecules travel through a membrane from high concentration to low concentration through the process of diffusion. No energy is expended, so it is called passive transport. When an embedded protein is involved in the process, it is called facilitated diffusion. Osmosis, the diffusion of water due to unequal solute concentrations, also requires no energy. In active transport, ATP is used to move molecules against the concentration gradient from low concentration to high concentration. The process uses carrier proteins pumping nutrients across the membrane. One of the most important pumps is the sodium-potassium pump, where sodium ions move out of the cell and potassium ions move into the cell, maintaining sufficient concentrations for living cells. Cells can manipulate their cell membranes to extend regions and form pockets. They can surround other particles and pinch off a vacuole containing the nutrients. This also works in reverse, where a secretory vesicle will fuse with the cell membrane and release its contents into the surrounding fluid.